Counting multiple DNA damage foci is important because each spot represents a biological event, and tracking several markers at once shows how these processes work together under disease or treatment. This is a dataset from one of Cytely users where cells were exposed to 2 concentrations of a DNA damage agent and stained for the nuclei and 2 types of foci, one in the red channel and one in the green channel. Here we've built a workflow to quantify both types of foci and the foci where the 2 markers overlap for each nucleus. The first step was to create a segmentation mask for the nuclei, and here I used segment cell nuclei. You can see that the mask is quite accurate, but sometimes we have these small holes within nuclei that we can correct for using fill holes. Then I proceeded to create a segmentation mask for the foci in the first channel. I applied an FFT high-pass filter to make the foci stand out more, and then I chose an overlapping background segmentation as the actual segmentation step. You can see here that sometimes when 2 foci are very close together, they get labeled as a single object. We can split those up using a peak intensity seeder and then flood fill right after that. I used a similar segmentation strategy for the foci in the second channel, and then I used keep overlaps to make sure to exclude those foci that are not within any nucleus. I also used keep overlaps to count the foci in the first channel that overlap with the foci in the second channel. Now we have 3 sets of objects, and we can group them all under the nuclei using this node. Here you can see the results. We use this first dot plot to create a gate to select the population of interest based on nuclei morphology. In these 3 plots, you can see the number of foci per nucleus sorted by foci type. We can also create a gate here to have a closer look. Here we plotted the sum area against the mean intensity of foci, again for both foci in the red channel and the green channel. These plots are useful if we want to create a gate on the population, but based on the characteristics of foci instead of the nuclei. Finally, in these bar charts, you can see that the mean number of foci for all foci types is higher in those cells that were exposed to DNA damaging agents, and then this response was dose dependent. You can summarize all these results and more by creating a table here, and you can also export them as a CSV file here.